Hello and good morning, everyone. Thank you for attending the AMCA Young Professionals October 2016 installment of our webinar series entitled The Biology of the Triatomines of South Central Texas and Assessment of the Risk They Pose for Autochthonous Chagas Disease Exposure, presented by Dr. Edward Wozniak. Before we begin today's presentation, we have a few housekeeping items to discuss. If you have difficulty with audio, you can call in to the meeting at any time using your phone. The number and access code are listed in the chat box on the left side of your screen. During the presentation, please submit questions for the presenters via chat. Questions will be answered at the end of the webinar during the Q&A session. There will be a quick survey at the conclusion of the webinar. If you have any comments on this presentation or an idea for a future webinar topic, please let us know. And now it's time to introduce today's speaker, Dr. Edward Wozniak. Dr. Edward Wozniak received his BS degree in zoology from Oregon State University and his Master's of Science in Veterinary Entomology from the University of Florida, where he studied the host immune response to soft tick salivary proteins relevant to epidemiology and ecology of African swine fever. He then attended the DVM PhD program at Purdue University to further expand his studies of vector-borne diseases. Upon completion of his PhD, Dr. Wozniak was awarded a 3-year NIH extramural postdoctoral fellowship in comparative pathology at the University of California, where he continued his study on tick-borne blood parasites. From 2008 to 2013, Dr. Wozniak wore many different hats, from a senior veterinarian and assistant professor at UTMB, completing an active tour of duty as a veterinary medical officer in the U.S. Public Health Service, and to also earning his Master's in Public Health at UT Public Health studying Chagas disease. He now serves as a subject matter expert on Chagas disease and other vector-borne pathogens and is an active research collaborator with several universities across the state of Texas. In addition to his work with DSHS, Dr. Wozniak serves as the Chief Public Health Officer in Texas State Guard Medical Brigade, where he holds the rank of Lieutenant Colonel and drills with the Preventative Medicine Unit of the 559th in the Medical Wing at Lackland Air Force Base. So, without further ado, I would like to give the floor to our presenter, Dr. Wozniak.
All right, Chagas disease. Chagas disease is a vector-borne protozoal infection. It is really found throughout the New World, from Mexico, Central America, and South America. This is a parasite that infects a wide range of hosts, including humans, dogs, and wildlife. There's a 3-stage progression. There's an acute phase, in which there can be local reaction and flu-like symptoms, which in some cases can be severe, resulting in myocarditis and meningoencephalitis. And there's an indeterminate phase, which is a latent period in which there is no symptoms at all. And that's the most common stage that we see humans in the studies that we've done. From there, about 30% of the people go into the chronic phase, which is where you see the severe pathology, and that's most typically cardiomyopathy leading to heart failure, and in some cases with some strains of T. cruzi, megacolon and megaesophagus. The vectors of this parasite are quite different from the mosquitoes you guys are normally used to working with. These are much larger insects. They are in the family Reduviidae, and you'll hear people say, Reduviidae bug bit me, so I'm exposed to Chagas disease. Well, that's not really true, because there's 2 different subfamilies to those insects. There's the Triatominae, which are your assassin bugs, which are predators of other insects, very stoutly built, as shown on the left there. Very, very stout legs, very stout mouth parts, and with a head that's usually angled up at about 45 degrees. These insects are very, very alert and sensitive. They're hard to approach. If you try to catch one, they'll oftentimes fly before you even get near them. On the right is the parasitic type, the Triatominae, which is the family of concern here for Chagas disease. These are blood-feeding insects. They live primarily as nest parasites. They have a compact body which is adapted for sliding into tight spaces, like between between boards in a house or into thatched roofs. They have a thin mouth part that folds very tightly up underneath, unlike the other one, which has more of a bobby pin conformation. And so morphologically, they're quite different. Well, Chagas disease exposure is a little different too. What we're looking at here is what we call Stercorarian transmission. The parasites actually shed in the watery feces of the insect. So the sooner the Triatomine defecates in relation to its feeding, the more dangerous the exposure, because those feces can get into the wound or get picked up and wiped into a mucous membrane. So if the feces are shed on the host, that's a more significant thing than if it's shed in the environment away from the host. Routes of infection include percutaneous transmission, where the parasite actually makes access through the bite wound inflicted by the bite. Mucous membrane exposure, in which the material gets onto a finger or something and gets spread into a mucous membrane like an eye or a mouth. Or through, in some cases, freshly contaminated foods. There was a case in Brazil where a triatomine got crushed in some juice, and a lot of people contracted the disease that way through mucous membrane exposure. Through bug ingestion themselves, little kids and animals mostly involved in this, and then artificially through transfusion and transplantation. Life cycle of this parasite is pretty complex. You have the flagellated stage, the triple mastigotes in the bloodstream, which are infectious for the kissing bugs, and then there's a amastigote stage, which is an intracellular stage, most oftentimes in muscle cells like heart muscle. And these will replicate and rupture the cell, emerge as a flagellated stage, in which they go through a number of different cyclical changes that really changes the position of the flagellum and the associated kinetoplast. And then these go on to triple mastigotes, which can infect the bug when they ingest blood from an infected host. Within the bug, the parasite colonizes the alimentary tract, and the infectious stage is shed, as I said, in the feces from the hindgut. The native distribution of Chagas disease, as I said earlier, is focused on the Americas. You got Mexico, Central America, South America, and parts of North America as well. So it's limited to the Western Hemisphere in terms of natural distribution. This parasite has been around for a very long time. Studies on paleoparasitology have demonstrated the presence of T. cruzi DNA in New World mummies as far back as 9,000 years ago. So it's nothing new for the New World. It's been here a very long time. The natural history of it is that the disease actually began several million years ago as an enzootic disease of wild animals. When humans ventured into these ecotopes and started bringing those animals into the house, some of the bug genera and species transitioned over to a domestic habitat and human association. The most notable species would be Wozniak prolixus and Triatoma infestans. Those are the most commonly seen ones in South America. Control initiatives, however, have recently decreased domestic transmission involving those species. Other triatomine species have recently emerged as major vectors of human transmission, and these are the more sylvatic bugs that establish transient infestations in homes. And as you can imagine, because they're environmentally based, controlling these is much more difficult than controlling a bug that is limited to just the internal human habitation, where you can apply pesticides. Some of the triatomines seem to adapt to living very well in human dwellings, just as our Triatoma infestans and prolixus have done in the past. Some of the other ones are now starting to emerge, show this tendency to be able to adapt to and live in and around human habitation. Heavy infestation of suitable homes is possible with both of these species, Rhodnius prolixus and Triatoma infestans. At least it's been historically before the control initiatives. And transient infestations with quite a few numbers of bugs are possible with some of these other emergent species, if you would. So the classic epidemiology that we see is a structure with lots of cracks and crevices in which these dorsal-ventrally compressed bugs can slide in and live. This really promotes bug survival because it gives them a good place to rest. And at night, when the human inhabitants are in there sleeping, the bugs will come in there and feed on them, and on goes the cycle. Vector transmission to humans are therefore related to bug habits and environmental factors both. There's sylvatic and synanthropic species of bugs, as I said earlier. The vector efficiency varies between species. There's host preferences, which some are more prone to feed on humans than others, and some of them have a different feeding to defecation interval. The longer that is, the less probable it would be for human transmission. House construction also comes into play. These guys thrive in porous structures. They're a nest parasite. They have to have a place to hang out, and they temporarily infest and bite the host. Because of that, you can use protective exclusionary features such as screens and solid walls to help protect against exposure to these insects, which in the United States is a standard of housing. So public health significance. This parasite is thought to affect about 11 million people in Latin America. It's a major cause of cardiovascular disease. Some of the things you see with this: arrhythmia, conduction abnormalities, dilatative cardiomyopathy and congestive heart failure, apical aneurysms, embolic disease involving stroke or pulmonary embolism, and in some cases, sudden death. Pathology, it causes what we call a non-viral infectious myocarditis. The heart often appears thin and dilated and very rounded, grossly. Microscopically, you'll find clusters, if you're very lucky, that is, of amastigotes in cardiac myocytes. In some cases, with a chronic infection, these are very difficult to find. You'll find the inflammatory cells between the myocytes and some necrosis, but sometimes the amastigotes themselves can be rather difficult to find in chronic infections. As I said earlier, some strains can cause megaesophagus and megacolon, and they do this by destroying the myenteric plexus, so those two whole visceral organs. This is a map of the different strains in the New World, and as you can see, in the United States, we have T. cruzi 1, which is a parasite well known to cause cardiac manifestations, and then T. cruzi type 4. And that one there has never been shown to infect people in the United States. We believe now that the type 4 in North America and South America are actually quite different. But this is an evolving science with characterizing these different strains. So Chagas in Texas, we certainly have T. cruzi present. We've had canine cases. Texas A&M identified a number of these back in 2009. There were losses of military working dogs that were being trained over at Lackland Air Force Base, identified in 2010 by Dr. Cropper and other people involved in that work. Been losses of Border Patrol dogs that I've heard about from local veterinarians that work with Border Patrol. There's been non-human primate cases in San Antonio and Bastrop. So with all of that, the question I have to ask is, what is the risk for human infection in South Central Texas? An ecologic model done in 2010 by UT Austin suggested that below 30 degrees north in Texas was in high-risk area for Chagas disease exposure, and that's based upon a number of different ecological factors. Texas certainly has a rich diversity of species of triatomines. And when you get into the biology of these triatomines, different species are really, really quite different when you get down to the fine levels. Here we have up to 5 different species in this general area. The only other area in the U.S. that comes even close to that would be Arizona, which also has 5 species. Traditional thought, though, is that triatomines native to the United States are all sylvatic species that don't readily invade houses or feed on people. They're thought they have prolonged feeding to defecation intervals that limit human exposure. Our higher standards of housing limits exposure, and indigenous T. cruzi strains are not pathogenic. Some of these have some truth to them, mainly the standard of housing. Some of these other things you'll see by the time we get to the end of this are myths. Recent observations and questions. There have been many reports of kissing bug exposure bites reported to this office in particular in Region 8, which is South Central Texas. So with that, I have to ask: Are indigenous bugs becoming more synanthropic? Are we getting in one of those cycles where some of them are adapting to live in human habitats? And if so, where are they residing and developing? What's their blood meal source? What's the rate of T. cruzi infection in these bugs? And what is the nature of human exposure in Texas? And are there any building design features that may be contributing to this colonization and exposure? My study objectives were to determine the species' relative abundance in the peridomestic habitat of South Central Texas. I really focused on that. To monitor native bugs for species-specific patterns of seasonal activity and changes in infection rate, and to monitor kissing bugs for species-specific tendencies to invade human habitations. Are there some species that stand out that are going in that direction, or seem to be, where others may not be? Determine the overall and species-specific infection rates, are the differences between them, and to monitor T. cruzi infection rates and vectors over the span of a calendar year. Does it change? Investigative methods: I collected, speciated, and tested kissing bugs using both passive and active surveillance tactics to get the bugs. I speciated the adults morphologically and used PCR in the immatures, and surveyed bugs for T. cruzi using both fecal spots and hindgut preparations, using a couple different Trypanosoma cruzi loci. So the surveillance, these were found to be, as other studies have indicated, very much secretive creatures of the night, very difficult to observe and collect. There's no real proven effective methods. Manual collection is oftentimes the best tactic. That's what's been found in South America as well. People have tried to design traps and without much success. Search underneath ground debris and animal nests is very productive for nymphs, but this is labor-intensive, slow, and can be dangerous, especially if you're in an area where there's a lot of venomous snakes. Looking underneath objects could easily get somebody exposed. Community specimens. I had people, when they found them, especially if they were bitten by them, submit them to this office, and I tried using attractants, both UV light and carbon dioxide being two of the primary ones. So field collection, we would set up a station like this where we have illuminated sheets with these UV bulbs and have CO2 along with it, and you just simply sit there and wait for the bugs to hone in on that. But then I started noticing that the bugs were very responsive to infrared radiation. If you have a kissing bug or an assassin bug, you're not sure which is which, put it in a plastic container, put your fingers up against it. As the heat radiates through, if it's a kissing bug, they will almost always try to bite through the plastic and get your fingers. Assassin bugs will not do that. So I thought, well, what about using CO2 from vehicles, a running vehicle, to give a heat signature along with the lights? Using that tactic, I was able to actually collect quite a few from some sites. In this one particular instance here, we got 61 bugs out of there, And had we stayed a little longer, that could have easily been 100, and that was in one evening here in South Central Texas. Other collection methods, community participation is very productive. Citizens, providers, medical and veterinary practitioners, and animal control officers all submitted bugs to this office for testing, many of which had bit the occupants of the house. So the results, I collected from sites all across this 28-county span in South Central Texas. I had a pretty good spread of collection sites. In terms of what was productive and what was not, UV and CO2, kind of productive. UV, CO2 in a car, a little bit more productive. Manual collection, very productive. And community participation, even more productive. But it's basically the same as manual. It's just you have more eyes out there looking for them. What I found were across the area, we had the 5 species that had been previously identified, although now not shown on here was another species that has become established here, at least in Uvalde County, but we won't get into that. Triatoma reticularia, sanguissiga, gerstaeckeri, protracta woodi, and indictiva have all been found across this area in varied numbers. If you look at my peridomestic data, one of the things that jumps off the page there is the number of gerstaeckeri collected in the peridomestic habitat versus all the other species. All the other species, 17, 15, 9, 10, over a 3-year period. That's pretty much what you'd get looking for triatomines across most parts of the country. Here we got, however, up to 400, and 357 of those were Triatoma gerstaeckeri. And over to the right, 64% of those were infected with Trypanosoma cruzi. So that's a lot of bugs and a lot of infected bugs right there, smack dab where there's human habitation. This is a pie chart showing the distribution of those different species. And as you can see, gerstaeckeri makes up by far the most of my collected species in the peridomestic habitat. Gerstaeckeri is our biggest and most obvious triatomine. It is a pretty large insect, very distinctly marked. There's no mistake in this guy, although some of the plant bugs and assassin bugs superficially resemble triatomines. This insect here is just clearly a triatomine. Once you get used to working with them, it's got all the features that are relatively easy to see on this large specimen. As with other triatomines, it's got a typical life cycle. There's eggs. There's 5 nymphal instars, and then the winged adults, male on the left, the female on the right. The female can be picked out by that little pointed abdomen. Eggs are percolated, laid in groups of 8 to 12. They don't stick to anything. They kind of fall to the ground. The nymphs are very secretive and seldom seen. You have to really get underneath materials and look for them, dig in nests and so forth. And the nymphs are very opportunistic feeders. They'll feed on almost any vertebrate blood available. It takes multiple feedings to progress from one nymphal stage to another. So this is a very slow process. To go from an N1 nymph up to adult can take more than a year in some cases, maybe even up to 2 years. Distribution of Triatoma gerstaeckeri is pretty limited in the United States, and it's really limited to Texas and adjacent New Mexico. That's the only places you will find it. And then from there it extends down into Mexico. If you look at the collection of gerstaeckeri, and this here is from the Texas A&M's map, this is based upon almost 2,000 specimens. Most of them are centered on the south central part of the state. That's where gerstaeckeri is found. This area consists of the Edwards Plateau, the South Texas brush country, and the up into the high plains a little bit in the southern extreme of the Panhandle. Within this area, Triatoma gerstaeckeri adults have a very distinctive period of activity. I start seeing them in late April. They peak out around June and then drop off dramatically over into August. Now, the nymphs are still out there in the environment, but this is where you find your winged adults, especially around houses. If I break that down further, you see here that in May seems to be the peak period, and it seems to taper off somewhat from as the season goes on and the weather gets hotter. If you look at the infection rate in gerstaeckeri and the tendency to invade homes, both of these seem to increase in the adults across the adult emergence peak. So as the season of activity goes on, these insects seem more prone to invade homes and are more infected, and a higher percentage of infection with T. cruzi. So what this suggests to me is a lot of these guys are not getting the parasite until they hit the adult stage, which makes sense. Bigger bug, to some extent anyway. Bigger bug, bigger looks for a bigger host, and that host may be more likely to be infected with T. cruzi. This is a PCR gel done by Baylor College of Medicine. This was a set of bugs that were found in a home. As you can see, most of them were infected. There's the TCZ band up on the top. And to make sure that the specimens were good, especially the community participation specimens, we always ran another reaction using Triatomine primers. So if we found Triatomine DNA was suitable for PCR, and we didn't get a parasite ban, we were reasonably sure that it wasn't just from degradation of the specimen, from mishandling. Triatomine bites, they look like, basically, as I tell a lot of people, like an exaggerated mosquito bite. You see a notable puncture in the center and a lot of erythema and a raised area around there. In some cases, you get this blanching of the tissue around it, and then spreading erythema around these sites. And some of this could be allergic reaction. Some of it could be, bacterial infection. But when I see that, I always start wondering about the possibility of T. cruzi involvement. These guys usually bite multiple times. This guy here was bitten several times, and all he found in the bug, or in terms of bugs and searching the bed, was one great big female. So multiple bites were inflicted on him in over just one night. This person here had multiple bites as the previous one, and one of them on the shoulder started to show that branching erythema around it, which was concerning for sure, because the bugs that were involved in this case were the ones that were shown on a PCR gel. There were a lot of them that were infected. So environmental factors associated with peridomestic triatomines across this area in particular. What kind of habitat are we looking at? Well, first of all, they're primarily around rural homes or edges of developments. I didn't find any right in the centers of any cities unless there was native habitat nearby. So clearly these are a species that has a sylvatic focus out there in the environment, and then they colonize homes opportunistically. They have to have that native habitat around to be around, generally speaking. So you can use that to your advantage by clearing such habitat away from your home. You can help to control those. They have a relatively short flight distance compared to many other insects, up to about 1 kilometer in the South American species. We really don't know for sure how far something like gerstaeckeri is able to fly, but we're assuming it's going to be about the same. Neotoma nests, wood rats, common around houses that were infested with triatomines. These are little triatomine factories. And you oftentimes find lots of nymphs developing in such a nest. They feed on the wood rats, and wood rats can be infected with T. cruzi. As a matter of fact, they are known to be infected with both TC type 4 and TC type 1 have been demonstrated in wood rats. So they're a pretty good host. Although, like I said earlier, most of my infected insects were adult stage insects, so I don't know if they picked them up from the wood rats or they picked them up from a bigger animal that they bit after leaving these little nymph factories, if you would. So where do they hang out in modern houses? We certainly don't have thatched roofs and stuff where the insects have abundant spaces, but we do have spaces nonetheless. This little crack underneath this door frame was a very productive area for triatomines. Siding like this and the gaps underneath it, I've watched triatomines disappear into gaps that were barely enough of a gap to discern it visually. They really can squinch down and go right up in there. Here's one going up underneath a piece of flashing. Here's one that just emerged from underneath some of this woodwork over here where there was a gap. And another one here that came out from underneath a piece of siding. Once again, look at the gap. It's very tight. So the question I ask is, are these bugs depositing eggs in this protective microhabitat, being the protective microhabitat being the external wall? Unlike primitive shelters, there's little communication, however, of the wall, external wall space with the inside of the home. We have solid walls, so that sort of limits their access to the inside. However, nymphal development would require a blood source. So what would they feed upon? Well, we know that rural homes are very susceptible to colonization by different vertebrates. Bats certainly go in there, mice, some lizard species, any of which could be fed upon by nymphs. We also recovered nymphs from inside Texas homes, and this is a very, very rare thing in the United States to find nymphal stages colonizing a house. Here's a set of 5 nymphs that were recovered from an individual's bed, and here's over on the right an N5 nymph that was recovered from a baby's crib. So we do see some level of what we would consider colonization of the home, where you actually have nymphal stages getting into the inside and feeding on the occupants of the house. There's another one here recovered from, once again, a crib, and it had bitten this child here on the leg. And you see this erythema about the size of a penny and a central puncture wound, typical triatomine bite. So observe species-specific differences. I mentioned that when you get down to the fine level of the biology of the insects, they're really quite different. Reticularia, protracta, and sanguisica, watching them around the UV light setups, they would fly to the UV illuminated sheets and land upon the sheets. They were active during the early evening and seldom found after 11 p.m. So after 11 p.m., you really didn't see them fly up, even in the dead of summer. When on homes, they were often found in lighted areas specifically, and they were sporadically encountered. Like I said, low numbers, maybe 15, 20 over a 3-year period is all I got. Gerstaekeri, on the other hand, never really flew directly to the lights. You'd hear them, but you'd rarely see them in flight. They were typically recovered from the ground around the attractant setup. So they would fly to the general area and finish the stalk to the CO2 source on the ground. These were also late-night species. The others dropped off at 11 p.m. I seldom found gerstaekeri earlier than 11 p.m. and oftentimes active well beyond 2 a.m. That's a late-night species, which is a very peculiar thing for a triatomine here in the United States. They're found in both lighted and dark areas on the exterior home, and they were observed using the cracks and crevices on a building. I did not observe that with the other species, although, once again, the numbers were very low compared to what I had for gerstaekeri. And they were frequently encountered in and around homes. feeding defecation interval. I've had gerstaekeri adults, and I've watched a lot of them defecate while feeding, which is a disturbing thing for sure, because that would put the parasite, if it's in the fecal material, right near the bite site. Here's one feeding on an artificial membrane, not the greatest picture in the world, but you can certainly see the defecation while in the act of feeding. Lackland Air Force Base, they had this dog develop an ulcerative lesion on the leg, initially thought to be a leishmania lesion, certainly could have been. They biopsied it, did PCR on it, and it was Trypanosoma cruzi. So what we're looking at here is a actual what we call a Chagoma, a beginning lesion associated with T. cruzi. And this is very good evidence of percutaneous transmission. So this bug had to have defecated near a site where it had fed and got the parasite into the wound. Evidence of pathogenicity in locally acquired T. cruzi, canine cases. We kept track of those here for a two-year period. 351 cases were reported in domestic dogs across the state of Texas from 2013 to 2014, and that's just the ones that were reported. 137 of those were symptomatic, and oftentimes they had evidence of heart failure, ascites, and sudden death. Those are very common features, especially in young dogs. Acute multifocal myocarditis with many intralesional amastigotes were seen, especially in young dogs. We had more cases in outdoor-housed Old World monkeys. Once again, lesions were centered on the heart, and lesions centered on the heart are very typical of T. cruzi type 1. So human cases, we had a kind of a lag in getting these reported, but they've been trickling in. Locally acquired cases, grand total for the 2013 and 2014 was 12. It's a small number, but when you consider the fact that previous studies from the 1930s up until now demonstrated only 7 nationwide, to find 12 in a 2-year period in the state of Texas is a pretty significant number, really. We also had a few imported cases and people that have traveled into known endemic areas, being south of the border, and the grand total between the two is 39 for that period of time. If you look at the distribution of human and animal cases, it kind of approximates that of Triatoma gerstaeckeri. There's our map from Texas A&M on the right, and showing where the gerstaeckeri were found, and the map on the left showing where the cases were found. Granted, there were cases outside of that area, but a lot of them were inside the range of gerstaeckeri. And like I said, gerstaeckeri is the bug that I usually found in and around homes. Not surprising that you would find human and animal cases where it exists. Baylor College of Medicine recently did a blood donor study. They compared Houston and San Antonio blood banks. Houston having sanguisica, but not gerstaeckeri as a common bug. There was 1 in 6,500 prospective blood donors were that tested positive between the two sites. In Houston, 17 were found, 36% of which were locally acquired. In San Antonio, where we have the gerstaeckeri, 73% of them locally acquired. So quite a difference there. And this data was generated by Dr. Melissa Garcia. So Trypanosoma cruzi and blood donation, it's definitely a public health concern. We certainly have local transmission. That previous slide clearly demonstrated that. So is it possible to have it in the blood supply? Well, the answer to that's going to have to be yes. We have local transmission. Current policy is to test once, and if cleared, people are cleared to donate. And that assumption is based upon there being no local transmission. So this is something that probably needs to be addressed further. Now that we have good evidence of local transmission, testing once really is not going to be adequate to protect the blood supply. So in summary, South Texas has endemic Trypanosoma cruzi and 5 indigenous strains of competent triatomine vectors, all I found all 5 to be infected with T. cruzi, but the gerstaeckeri being the dominant one to be in its peridomestic habitat, to be highly infected, and often found in or on homes. And usually this species involved in human bites, at least in this region. Triatoma gerstaeckeri ecology may be changing. Just as we've seen with some of the other triatomines over time, this one may be evolving to be able to colonize and take advantage of the human blood that's available in new areas as they expand cities and more people move into these rural habitats. T. cruzi infection rate and tendency for adult stage gerstaeckeri to invade homes both increase over the period of the seasonal emergence of the adult stages, and those were both statistically significant values there. Parasite-induced behavioral change is possible. With some species of triatomines, T. cruzi has been shown to actively change their behavior and make them more prone to bite and so forth. We don't know if that happens in gerstaeckeri, but it's something worth maybe investigating. The genetics of our bugs too. Are there differences between the sylvatic gerstaeckeri and the ones that are coming up and living around houses? We don't know. Certainly been found with other species in Mexico. It could be happening here as well. The variable feeding to defecation interval with gerstaeckeri adults, they oftentimes defecate while or shortly after feeding. With something like Sanguisica, although I haven't fed a lot of them, it seemed to be much slower than that. I didn't really observe them defecate while feeding. Infection with indigenous strains of T. cruzi can certainly cause pathology, at least in the dogs and non-human primates. And in some of the human cases, they've demonstrated EKG findings consistent with T. cruzi infection. So our strains here are definitely pathogenic and dangerous. There's been a reluctance of medical providers to test or even consider T. cruzi as a likely source of cardiac manifestations, and this has probably contributed to major underreporting. CDC put on a grant that was obtained by UT School of Public Health to address this, and there's ongoing physician education programs as we speak, trying to change and bring it to their attention that this parasite is here. It can be locally acquired, and to consider it in your differential diagnoses. So how does Chagas disease compare here versus the tropics? The classic epidemiology being these thatched roof huts. Here we have solid walls inside, so really probably what happens is the rate of exposure of a person here would be intermittent, and whereas under your primitive conditions like this, it's probably almost on a daily basis, where there's lots of bugs in a house and lots of exposure. Here it happens, but it's more intermittent. So South Texas, to me, is very similar to parts of Mexico in terms of Chagas disease ecology and epidemiology. As I mentioned, we have the bugs which are environmentally based that opportunistically colonize houses, and you get temporary infestations of them. It's very similar to what we see in the Yucatan with Triatoma dimidiata. What can be done? It definitely needs something, a One Health approach and integrated pest management to control this. These are complicated life cycles, environmentally based insects, very difficult to control. So surveillance, I suggest looking for fecal spots. Sometimes you'll find them on bed sheets or pillows, sudden appearance of a black spot, waking up with a large insect bite. I advise people to tear apart the bed and look for bugs, and oftentimes they do find them. To collect and submit possible specimens, I encourage that. Oftentimes now I ask them to take a picture if they're not sure on the species, and if I can rule it out as being a plant bug or something, I won't have them sent in. But if it's a triatomine, I'll ask them to submit it. To look for eggs and nymphs and nymphal exuviae, that's getting a little bit more tedious, but you can certainly find those around an infested bed or around an infested pet sleeping area in a house, especially if you use sticky traps. Local preventive and control strategies, IPM tactics, regular surveillance, crack and crevice treatment of the house seems to help a lot with keeping them out of the house, even if you have an external wall infestation. Controlling vertebrate pests around, clearing vegetation back to eliminating the native habitat that's close at home helps. Keeping the areas clean. You want to avoid clusters of hair like this. This is a nice interstitial environment in which nymphs could hang out and feed up on your dog. You want to keep that vacuumed up and out of there. So another question we have is, does a dog, family dog, which can definitely be infected, be a zoonotic reservoir? It's theoretically possible, especially if you have a lot of material around that can contribute to long-term nymph survival. Eventually, those nymphs will find their way probably over to the human habitants as well. So exposed residents, a very slow time course to develop Chagas disease. We're talking years between exposure and the development of chronic manifestations. During the acute phase, you can do positive. You can do smears, you can do a PCR, or you can do culture to detect the parasite. After about 8 weeks, then it goes into the indeterminate or chronic phase, and you can detect it with serology. So yet it's hard, but sometimes you have to tell people, well, you really need to wait a few more weeks before you collect a specimen. In the meantime, they're sitting there worried about it, but the serology takes a while to come up. Antiparasitic treatment, the only effective treatments that we have that are available only through CDC. So the way things work now is people will be tested. They can be tested in a commercial lab. That's a preliminary result. If they test positive, CDC will do a confirmatory test. If it tests positive on CDC's test, then that person or the person's physician will be eligible to be put on the CDC IRB, because these aren't under they're non-FDA approved products, so they need to be used under an IRB, and CDC has an IRB they will add the physician to, and then the person can then be treated by that physician. That I'll open it up to any questions. And sorry for the technical difficulties in the beginning. Tried to make up some time, and I need to acknowledge all of these people who were involved in this work with me. I certainly didn't do it alone, and it's been a great study, and it's certainly ongoing. And we have many spin-off studies that are stemming from this. Thank you.
Thank you so much, Dr. Wozniak. That was very, very interesting. And now we'll begin the discussion portion of our webinar. So if anyone has any questions, please submit them in the chat box on the left, and we'll get to them one by one. I actually have a couple questions.
Sure.
I'll start if no one else has any. So with the kind of lack of housing structure for homeless populations in the state of Texas or anywhere else in the United States, is there a big problem or possibility of risk with homeless populations in the United States?
Well, certainly they're being out in the environment. They're certainly highly susceptible to being, bitten by the kissing bugs, and if they tend to stay in the same area, create sort of a home, if you would, but an accessible home. I mean, that I would say would be likely to become colonized by the kissing bugs. Both Texas A&M and I think Baylor have done some work with homeless people, and they have found seropositive individuals in their surveys. I know from talking with some of these other investigators. I'm not sure how that compares with other populations. So Melissa Garcia may be a good person to ask at Baylor.
Mm-hmm. I imagine it would be a difficult population to survey, and there might also be some HIPAA kind of
absolutely
problems with that. Yeah.
And they've got to be willing to to be tested, and then they would have to have a physician to treat them if they chose to be treated, and so it's a long-term commitment there. They have to keep on going back on a regular basis for the administration of those drugs, even though they're oral, they're orally administered pills. They still need to be done under the watch of a physician because it's an investigational treatment.
Right. I have another question.
Sure.
For individuals who are sending in specimens to your program, how dangerous is it for them to pick up the insects, or what if they touch them? How safe is that, and how do you advise people when they're sending in insects?
I usually advise them to just kind of use a large mouth container and kind of sweep it into there, or place the container over them and sort of manipulate them into the container so they don't pick it up with their hands. Although I've picked up hundreds of them myself. I've never really had one try to bite, and I've never had one. Well, I take that back. I did have one defecate one time, and I immediately washed that area off with soap and water and then alcohol. I didn't have any open wounds on the area, but nonetheless, I wanted to make sure I got rid of any parasite material that's there. So yeah, that's always a risk. But if the bugs are in their home anyway, they're probably getting exposed to them while they're sleeping. So in terms of relative risk, catching them is a lot less dangerous than sleeping in the house where they are, because they really, if a person's moving around, they're not going to bite. They're very, very sensitive to movement. You start moving around, they detach and take off, which is probably why you get multiple bites from one insect on a person. Anytime that person moves, they would disconnect and then come back when they're stationary again.
If someone did send them in, how do you preserve the bugs? With ethanol, you freeze them, or are they supposed to be alive?
Well, in my case, I usually had, if they had live bugs, I had them submit them live. I told them to just put some paper in the container, fold it like a piece of, like a coffee filter, so the insect have something to hang on to, and go ahead and send it live. A&M asked them to freeze them, and they were sending them frozen. I had people do that. Problem with that is when they pull them out of the freezer, then you have all the ice crystal damage, and a lot of times it's moist. So if it spends much time in the mail, which oftentimes they do, you'll have degradation of the insect. I've had some of them that were just like a gob of stinky goo, and those, testing that would be, questionable at best. So what I usually did is, if they told me that the insect was dead and it was still moist, where it could grow fungi or bacteria, I usually had them put them in alcohol and submit them in alcohol, because at least that stops the degradation, and it's still, your DNA is still very good, very good with alcohol preserved specimens.
Right. Another question someone submitted was, what pesticides are effective against kissing bugs?
Well, all literature indicates that, because they have a relatively slow generation time, that they haven't developed a lot of insecticide resistance. So most of the synthetic pyrethroids are highly effective against them. Permethrin, cypermethrin, any of those seem to work pretty well. I use cypermethrin in one of my houses I lived in previously, where we had a lot of bugs in the external walls. I didn't want them. I wanted to collect them and watch them and stuff, so I didn't want to do anything to disturb them outside, but I certainly didn't want them getting inside. I kept the wall floor junctions and around windows and doors treated with that insecticide, and it did very, very good at keeping them out. I got a few in there that probably flew over the borders of the insecticide, but even if they do that, with time, like any other insect, they're going to go to that wall floor junction and get into the pesticide, and you will get them. Hopefully you get them before they bite you, but it seemed to help a lot. I didn't have a real problem. I could find loads of them on the outside, but very few on the inside.
Okay, that's really that's good to know. So if anyone else has any other questions, we'll wait just a few seconds if you want to type them into the chat box. If you would rather submit a question privately, you can send them to our Gmail account, which I am typing into the chat box right now. So, or if anyone has any comments, you can send them to our email. And I don't see any more questions, so we can kind of wrap up. So I would absolutely like to thank Dr. Wozniak for speaking with us today, especially everyone for attending and sticking with us through our technical difficulties. Very much appreciate that. So remember, there is a short survey at the end of this presentation if you so wish to fill that out. A recording of this presentation will also be available on our website and Facebook group page, and we will get that up as soon as possible. So this concludes our webinar, and thank you again, everyone, for joining us.
Which web page are you talking about for the?
Oh, the American Mosquito Control Young Professionals. We have a web page where we have an archive of all of our webinars.
Would you do me a favor and send me a link to it?
Yeah.
Because I have some people I think that probably want to see it that weren't able to. If it's going to be available, that so they can go back and I didn't realize they're going to record it and it would be available.
Yes, sir. And that was a great segue to promote our website. So thank you.
Yeah.
All right. So thank you, everybody.
Hey, thanks. I'm going to go ahead and log out of it.
Yes, sir.